Still in Chapter 2, Natural Killer Cells versus NKT cells. Natural killer cells, as I mentioned in the earlier little clip, kind of count the number of MHC molecules on the surface. There are several different kinds of viruses that will try to downregulate the production of MHC class 1 molecules in a cell that they're infecting. They're trying to lower the production of those MHCs so that they're less likely to express foreign or bad antigens on the surface of those cells. Well, normal cytotoxic T cells will only go by and they'll read the few MHC1 molecules on their surface and they'll say, okay, you're okay. But a natural killer cell comes in and it can kind of, they look and they say there's not enough of these MHC receptors on the surface. That means that there's something going on inside and they can induce cell death. They release cytotoxic chemicals which cause the cell to go through kind of a programmed cell death. NKT cells are kind of one of the hottest areas in cellular immunology that's out there right now. And they have characteristics of both the cytotoxic T cells and of the NK cells. They have the ability to recognize antibodies that are bound to receptors. The FC region of the antibody will bind to these cells and stimulate release. They can cause large amounts of cytokine release. They can downregulate or upregulate the immune system, and for some reason they believe they're extremely involved in asthma. So they think that maybe these cells are overreacting in patients with full-blown asthma, which means that if we can come up with a way of regulating these regulatory NKT cells, then we might have an effective treatment for asthma patients that doesn't suppress the rest of the immune system. I'm not going to do as thorough a job on the rest of this chapter because, I mean, you are college students and you can read. Be sure to know the differences between negative and positive selection. Know some of these terms in the stem cells, clinical uses and potential. Autologous, synergenic, allogenic, graft versus host disease, gene therapy. All those are terms that are kind of important. When you look at the chapter on bone marrow environment, just realize that these cells that are growing and dividing and learning inside the bone marrow, it's not just them being genetically prone to it. They have to have the right stimulation to produce them, to produce the right kinds of cells. So the yellow fat that's found in bone marrow is part of the environment that is necessary for these cells to develop. If you get stem cells, the bone marrow is the place to do it. So these cells are squeezed through small capillaries. They're exposed to the right receptors, which cause them to develop in a certain direction. And that's one way that you can control how many of each blood cell type that you have is by controlling the environment in which they develop in. Look at the structure of the thymus gland just briefly. The thymus gland is kind of above the heart. It's a double-lobed organ. They're finding now that some animals have thymic glands in other areas. So if you were to remove the thymus, there are other thymic organs in the body that can maybe pick up and do a little bit of the T cell training. Just kind of get an idea of where the lymph nodes are in the body and what the structure and function of all of that is. Now, the second thymus is a little section on them finding that secondary thymus in mice, and they're also finding it in species like the koala and the kangaroo. I don't think anybody's ever studied the possum, but it might be something that could be looked at. So read through the rest of this chapter, learn what double negative and double positive are, and if you have questions about it, I can make another audio clip that's on there. The lymphatic system helps to remove interstitial fluids that have leaked out of the capillaries and moves it back into the circulatory system, but it's also like a singles bar for these white blood cells. It's where they hang out. It's where they come into contact with each other. So you're much more likely to have the right white blood cells come into contact with one another if they hang out in the lymph nodes that are near where the infection was isolated or found. Let's see. I'm not going to make you memorize the intimate structure of medulla and outer cortex and all of those regions of the lymph nodes. Just know that there are specific regions within the lymph nodes that carry out certain functions. MALT is mucosal associated lymphoid tissue. GALT is gut associated lymphoid tissue. You have non-node lymphatic tissue that's found in the intestinal tract, in the lining of every mucosal lining in the body. And this is where some of these blood cells are going to hang out, where you're going to get strong immune responses develop. It kind of puts geographic regions on where the different types of cells are going to work. Look at Peyer's patches, where mucus is produced, how antibodies cross the membrane. Figure 2-12 is one that you really need to look at. Those M cells are very important for transporting antibodies across the membrane. So the antibodies are produced inside, but they're still secreted into the surfactant of the lungs. In the nasal excretions, your earwax has certain antibodies in there. So how do they get across those basement membranes? How do they get across the epithelium? That will actually give you a good look. I don't have time to talk about developmental or evolutionary immune responses. You can just glance over it, learn what the bursa of fabricus is. That was where it was first found. A thymus-like organ was found in chickens. And a guy from Mississippi State was the one who discovered that. See if you can find out who he is. The summary in this new book are really good, and there's lots of really good animations on the useful websites link on page 61. If you have specific questions, you can post them on Blackboard or shoot them to me, and I'll try to answer in an audio clip so that everybody gets a chance to see what the questions were. All right, next is Chapter 3.